Good morning, everybody. Mostly familiar faces from yesterday and some new ones. I will be telling you about all the things that can go wrong, like before you even send your samples out for sequencing or out for proteome analysis, mass spectronomy, or whatever kind of metaomics technique you have decided to do. I will tell a little bit about who I am and why I talk about these things. I actually pretty much skipped my presentation yesterday, so I will promise to do better work today. I haven't done metagenomics. My background is in laboratory-based microbiology, and especially in environmental microbiology. So I actually did my PhD at the Faculty of Agriculture and Forestry in University of Helsinki. Defended my thesis about 5 years ago from the research group of Kristina Lindström. And I'm really a soil microbiologist, so I study one of the maybe most difficult and definitely one of the most complex and diverse microbiomes that there is in the entire world. I've been studying a lot hydrocarbon polluted soils, using a lot of community fingerprinting techniques that are actually very good for testing and ensuring different quality aspects of your research. Then I did one postdoc in Lahti at the section of environmental ecology, working for Martin Romanchuk. And I got a 1-year personal postdoc pool funding to carry out a research project taking samples from Iceland to study desert microbiomes. And then for 10 months I visited the research group of Noah Ferrer at University of Colorado in Boulder. So there I actually first learned about MySeq sequencing and really a high throughput microbiome analysis. That lab is probably, at least in environmental microbiology, one of the best known or most famous environmental microbiome analyzing labs. Currently I work for Marja Tiirora at the University of Jyväskylä. She has an ERC, an EU-funded project for development of a novel method called microRIP. But we also do lots of different collaborations, for example on plant walls. If you have heard about a product called Naturvention or NAVA, green walls. We study chlorophenols and lots of other things. And in Marja's group, and at the University of Jyväskylä, we do a lot of ion torrent sequencing. So there I actually do sequencing myself as well, and we have the equipment there. But yeah, my background is in soil research, and so will be many of my examples. And of course, like most of you are probably not studying soils, and for example, our Norwegian guests are studying marine samples, I've understood. So it would be really interesting to hear about your comments and viewpoints and like how your samples and cases might resemble some things that we have stumbled on. So I would very much hope this to be a little bit more interactive, not just lecturing. So if you have any like burning comments during the lecture, please feel free to share them. We should then probably use this box so that also your questions or comments will be recorded, because I think they are very important. Like the lecture entire will be recorded and should be recorded. And also at the end we will have a question session, so there I can either repeat your question to the microphone or I can just throw the box to you if you have a longer question or comment. But anyway, the content of the first about 60 minutes. I will be discussing about experimental design and most importantly replication. I will tell what can go wrong, like give some examples, cautionary examples about how things can go wrong with sampling and sample storage. And I will also present examples of how different things in nucleic acid extraction can affect your recovered community. So when we do science, not research, we have been reminded that we should think that we are scientists. We should have scientific questions and scientific thinking. The first thing that you should think is, what is your question? Like, not that I have 5,000 euros that I can use, or 10,000 euros that I can use on metagenomics that my PI promised for me, or that I got from some foundation. But really think about, what is your question? What is something that you want to answer? Something that has not been answered, and something that should be of interest to a wider audience. And then when you have your question, then you should decide what method you could use to answer that question, and which method you can afford to use to answer that question. And here I've listed some prices for, I think, outsourced metaomics sample runs. These are mostly prices that I heard at the Soil Metagenomics meeting in Braunschweig in December. So they are pretty recent, but of course the prices may differ. But this probably gives you the idea of what the prices are per sample. And then think about, like, what are the questions you can answer with those techniques. For amplicon sequencing, you can answer which mostly known organisms are present in your sample. I have known there because our primers need to match those organisms. If it's something that no one has seen before, maybe it's because primer bias or primer mismatch, so maybe our current primers aren't even able to find something like really unique. For metagenomes, you would recognize, of course, also the 16S genes, so which organisms are present in the highest numbers, because you can rarely, like, fully thoroughly sample your community. And of course, what we would like to hear or know about is that which functions are present. Also there I've written known, because, well, for example, from a soil metagenome, typically something like from 10 to 20% of the genes that you identify are from protein coding or some kind of functional genes. You can actually identify and tell what those genes do. And that's one thing why I actually haven't been horribly excited about metagenomics in soil environment, because who knows what the 80% is doing. Like, I don't know if all the interesting things are there. So obviously we still need really lots of work on developing the reference databases. And actually maybe this has already improved. These examples are from some years ago, and the field is going forward incredibly fast. So we'll hear more about what you can identify nowadays. For metatranscriptome, you sequence all RNA. So unless you get rid of your ribosomal RNA, which is definitely the way biggest pool, most of your RNA sequences will be just telling you which organisms are alive in your sample. And as well, for the messenger RNA, you will hear, or you will learn about which transcribed functions are present. So what is the transcribed metabolic capability of the community of interest? And for metaproteomes, I've heard that typically you identify from 100 to maybe 1,000 proteins. So think about that if you want to do metaproteomics. It's definitely not like very deep sampling depth. I think 1,000 proteins is at least according to the soil metagenomics meeting or presentations there, 1,000 proteins is already pretty good from one sample. So again, you will learn about what are the most abundant translated functions. It doesn't mean that the enzymes are actually active in your environment, but the proteins are there. And also, maybe you will learn something about structural proteins and what living organisms are present. So if the prices frighten you, and if you think that, well, maybe I cannot afford some of the fancier analysis, it is good to remember that 16S data, the amplicon sequencing, is used for lots of things. It does give you an indication of what functions are found in your sample, just based on the identities of the prokaryotes. So you can search literature, and you can use tools such as PyCrust, where you also need a reference database of fully sequenced genomes. Also, when comparing your samples based on 16S or amplicon sequencing data and based on metagenome data, the similarities or dissimilarities of samples are often very similar. So for example, in this paper from Noah Fierro's group from 2012, they found out that the beta diversity patterns, the principal coordinate analysis from 16S and based on metagenomes were nearly identical. And also here I've quoted that at that time they were only able to identify or annotate from 13 to 23% of the soil metagenome sequences. When you start to design your experiment and your sampling, one of the big important words and concept is metadata. Metadata or environmental data or contextual data or like explanatory variables or explanatory categories. So before you even start your experiment, you probably know your system. I would very much recommend you to go to see the MIMARC's guidelines, this minimum information about a marker gene sequence. Because those data that are required for your system, for your biome, you need to store in the sequence databases upon archiving or storing your sequences. When you publish, so thing is that you need to have this data to be able to publish your sequences, to be able to publish a manuscript or an article. It really is required by reviewers. So it's a good idea to make sure that you can collect all these even before taking your sample. And note also that, for example, soil pH or lots of different things, they can be very wide heterogeneity, and things can change actually even pretty fast, some things, after sampling. So it would really be recommended to do really all the analysis from exactly the same sample. And of course this will produce some preservation challenges. So if you, for example, need to collect, say, 2 kilograms or 5 liters of your original material, then how will you store it in the field appropriately or in the laboratory? I will get back to this a little bit later. Also, you should think about how representative the samples you analyze are for your system or for your biome. Because, like, you analyze samples, but of course you want to study the system. So you should think about how well your samples really represent your studied biome, and how well they represent the diversity or the heterogeneity in your studied biome. If your system is very heterogeneous, you should have more samples. If the response that you want to see in an experimental setting is very heterogeneous, you need more samples. So it would not be a bad idea to calculate. There are actually very simple, even like online calculators for power analysis, or just like discuss with a statistician to make sure that you have enough samples to see the size of differences that you want to see. If you are planning to do something that is pretty expensive, that could be, say, 1,000 or 2,000 euros per sample, what I would do is that I would collect at least 6 samples from my system, 6 replicate samples, biological replicates. And then I would describe them or characterize them with some cheaper method. For example, amplicon sequencing. So then I would be able to, maybe based on some like preliminary data, like see which of those 6 samples are maybe the 3 that are the centroid samples, or the most diverse samples, depending on what you want, but how that best characterize your system. Because if you end up doing, for example, metagenomics, and then you have maybe only 2 replicate samples, and they are really different, you have absolutely no idea which one is the right one. Or let's say that you have 3 samples, from which 2 are very similar, and then the 3rd biological replicate is really different. You have no idea if that heterogeneity is true to your system, or if it's just because someone sneezed. I think metagenomics would be a horribly expensive way to find out that someone sneezed. This was a catchy title of an opinion paper by Jim Prosser, who's, I would say, I'm a big fan of his. He's a soil microbiologist, a senior soil microbiologist who very much like talks about these things that I have been talking about now, about replication, experimental science, and scientific thinking. If you're interested in these kind of things, like, just find his papers, really nice, and also usually very entertaining papers and talks. But yeah, already what I talked about a little bit during the previous slide. Replicate or lie. Like, if you don't have replicates, you really cannot tell about your system. So the biological replicates should ensure representativity, that your samples really represent your system. And I would also really encourage to do at least some technical replicates. And to me, technical replicates would start from extraction, from nucleic acid extraction, in the case of DNA or RNA-based analysis. So just duplicate or triplicate nucleic acid extractions from exactly the same sample. Because from those you learn what is really the technical reproducibility of your entire analysis pipeline. And that helps you to interpret how important the differences that you see then actually are. Could it be just some technical bias, or is it so sizable and also so logical that it could be something biological? I also inserted here this, I think, hilarious quote from Environmental Microbiology journal's reviewers' quotes from 2012, that one sample, it's not enough unless it's from Mars. In that case, it could be interesting enough. Nothing else, one sample, not enough. So recommendation, this is actually from the soil proteomics talk, but I think it very well fits to other metaomics or amplicon analysis as well. So it would be recommended to do from 3 to 6 biological replicates, and I think it would be really interesting to see 3 replicates, 3 technical replicates, at least. Of course not for all of your samples, that would be wasting money and wasting resources, because statistics you do on biological replicates, but at least like for 1 or 2 samples to see the technical reproducibility of your analysis. Okay, so we have our question. We have decided our technique. Then let's proceed with sampling and sample storage. Here I was thinking of just sharing 2 cautionary tales, like how can things go wrong. So what I really strongly recommend, if you are working with a system that you or maybe not even anyone in your group has worked before. At least environmental microbiologists do that all the time. We jump from one system to another. So if at all possible, then testing the sampling and all procedures before you have your precious unique samples would be very recommended. And in my experience, like Murphy's law really works here. So everything that can go wrong will go wrong. And I have seen that so many times. So I said that my background is very much in bioremediation or contaminated soil research. We started an experiment with I think something should have had like pretty nice experimental setup and statistical design. We had 12 blocks. Block always had 2 experimental plots, 2 times 2 meters. So 12 blocks, and in each of the blocks we randomized the side of the treatment and the standard or control. What we were actually looking for is rhizoremediation or plant-assisted bioremediation of contaminated soil with a leguminous plant. So to every other plot we planted Galega orientalis or Su, the seeds. And every other plot was otherwise treated exactly the same way, but we didn't sow the seeds there or plant the seeds of the plant. When you do experiments like this, what you want to do is you want to start your treatment or your experiments, in our case, like throw the seeds in the plots. And then right after that, before there is like anything, like biologically time for anything to happen, you take the first samples, the initiation samples. So of course, just sowing the seeds should not make any difference. We would want to see exactly the same results from control and treatment blocks. It was a pretty extensive sampling scheme because we wanted that our composite soil sample really represented the entire experimental plot. We had this kind of scheme. The plot is here, and we had this systemized drilling scheme for soil cores down to 20 centimeters depth. So like here, so that we know that we would pretty well always characterize the entire plot. It was a lot of soil cores, or a lot of drills, and it was like hard, dry soil. Actually, the total number of soil cores was 384. So obviously no one person can do that, and not even two people. So we had four girls, 4 ladies, and to minimize cross-contamination between the test plots and the control plots, even though we didn't expect, of course, there to be any difference, but to minimize it, one pair took the samples from the test plots and another from the control plots. We had exactly the same kind of soil cores or drills, exactly the same kind of like advice or protocol, like, you know, just core it down to 20 centimeters and then just drop it in this bowl, and then without mixing we store them all in a plastic bag and take it to the lab and sequence during the same day. Not sequence during the same day, of course, but sieve during the same day, and then store frozen. It was a tough day. The sampling and the travel to the field and back took something like 8 hours or 10. And then the sieving and sample storage and all the things that you wanted to do during the same day took like 8 more hours. So 16 hours working day. When we were finished at midnight, we were like joking, like, wow, let's do this again next week. Like, you know, like really, really tough sampling day. Well, it ended up that we actually had to. So, this is results from soil respiration. So basically just the production of CO2 from soil. It's an indicator of the organic matter in soil. It's an indicator of the activity of microbial community in the soil. Even if you haven't done soil respiration ever, you probably understand that at the initiation moment of the experiment, you should not have any systematic difference between your treatment and your control blocks. So when we saw those results, we were like, wow, like what could it be? Like, what on earth caused this? And really the only difference was that one pair was sampling the other plots and another pair was sampling the other plots. And even though we were doing it in exactly the same way, I think the difference was that the other pair used hammers to push, or these kind of like rubber hammers, to push the soil core down to the depth of 20 centimeters. And we didn't have hammers for everybody. So the other pair used more like just like push it with your feet or try to sit on the core to get it down to 20 centimeters depth. And that was really the only thing that we could figure out caused this. So then, what we had to do is that we redid it in I think 2 or 3, yeah, in 2 weeks. So what we then did is that all 4 women took samples from every single plot, so that each of us, there were I think 12 different samples here. So each of us always took 3 cores from each plot. And then we got perfectly, beautifully even results for the control and treatment blocks. So just like a cautionary tale, things that you would never think affect your sampling or your samples, they can. Another research, actually an article about spatial distribution of soil characteristics. We have another article about the effect of sample storage. But here, with this experiment, we ended up seeing also the effect of sample storage. So in this experiment or research, we took 50 samples from the same like soil mineral type, but 3 land uses. We had adjacent or very close by field and meadow and forest. And the sampling scheme was like this. We had grid samples, so again we had like very carefully systemized sampling. Grids, like regularly interspaced grid points. And we had these randomized point. So every grid point to have like a different like variation of different distance classes. We had randomized the points so that we randomized the direction and the distance of a randomized point for each grid point. So this it was like systematically randomized, but you ended up with this like random looking scattered pattern. But of course they were taking at the same time. So when you first took the grid point and then you sampled the randomized or the near point, or these small soil cores, maybe top 10 centimeters. Of course, same cores, same people sampling, everything done the same way. So those 2 sample or sampling point classes shouldn't really bear any meaning other than that you get these like intermediate like distance classes, distances between the sampling points. This I will actually skip. The emphasized results you can read from that paper if you really want to. But what we found out then, when we were looking at the bacterial communities in those samples, we used a community fingerprinting method called LHPCR. That's a very well like reproducible, very quick and fast and easy, and also very cheap method for just seeing how similar or dissimilar your bacterial communities are between your samples. So LHPCR, community fingerprinting for bacterial 16S. And then here I show an ordination, a 3-dimensional ordination of what the results look like. In an ordination, the samples that are more similar to each other cluster closer to each other, and samples that are different are further apart in the ordination. So here I've shown the grid samples and the randomized points or randomized samples with those different shades for field, for meadow, and for forest. For field and for forest, there's like a random scatter that you should expect. For meadow, we see 2 main groups, 2 big clusters. So the grid points and the randomized points cluster separately. You can actually also see it here in a different kind of visualization, a dendrogram of the samples. For meadow, you see them cluster separately. So this is not something that like was caused by the ordination. And we were really surprised to see this. And we actually still like a little bit argue and negotiate like why was this? Because we didn't see the same pattern for field or for forest. So obviously there wasn't something like really systematically wrong in our experiment design. The only thing that differed for this was that it was a heck of a lot of soil samples that you needed to sieve and mix and then store frozen. And we couldn't do it. We had 3 or 4 people. We couldn't do it even in one day. Because you want to clean up the soil sieve between the samples and avoid cross-contamination, and all these things take a lot of time. So all the field samples we were able to process on the next day. So the small soil cores waited in these mini grip plastic bags, tightly sealed in the fridge for just one night. For the meadow, we were able to process half of the samples on the next day. And the other half, the near points, had to wait in those tightly sealed bags for 2 nights. And for all the forest samples, all of them were processed 2 days after sampling. The only thing we could think that differentiates this, whether your samples wait in the fridge for over 1 or 2 nights. In the fridge, like really enclosed bags, without breaking the soil cores. So that's how sensitive your microbial communities can be, even with this kind of not so sensitive methods. So really think about the duration of sample storage before you actually store them long term. I think long-term storage is actually much easier. Because for DNA, we found out that if you store your samples even at -80, even at -20 degrees, it really didn't change the microbial community profiles at all, compared to samples that were processed fresh, like right after sampling. For RNA, I think everybody recommends snap freezing in liquid nitrogen. Also some chemicals can be used, and other people recommend other, and other people recommend other. Usually it seems to maybe depend on the matrix very much. But with this like snap freezing in liquid nitrogen, just think that you should do all the analysis from the same aliquoted sample. So it would be good, for example, to first collect like a bigger mass of material or bigger volume of material in the field, and then mix it all well, and then snap freeze a small proportion of that well mixed sample for RNA analysis, and then collect the rest of the mass or volume and store it appropriately for the other analysis. Also, nucleic acid extraction or methods for that can have huge, like small things can have huge effect on your recovered communities. One actually pretty interesting thing is that according to this publication from December 2016, so last December, at least in soils, maybe not so much in waters, but soils and most likely also in sediments, there's a lot of relic DNA. A lot of DNA that is coming from cells that are not anymore alive. So again, a publication from the Fearer group, who found that 40% of prokaryotic and fungal DNA was either extracellular, so from cells that are already degraded, and DNA is stored attached to soil minerals, or from so-called zombie cells. So they may look like intact cells, but the integrity of the cell membrane is compromised, so they are not living anymore. They also found out that this extracellular or DNA from non-living organisms inflated the observed diversity of both fungi and prokaryotes by up to 55%. So that's pretty much. It's a good question, like if we study DNA, like does it actually, at least in the context of soil, really tell more about history than about the present moment? But if you study DNA, there are also lots of, like, of course, it is easier, it is in some sense more maybe robust or more even. And there are like other justifications to do that. Then you have to decide how to extract your DNA. Again, we're testing different extraction methods for hydrocarbon or oil contaminated soil. We compared different commonly used protocols or kits. The fast DNA spin kit, MoBio, the regular soil kit, and the max kit, where you can use up to 10 grams of soil. And we had the manual Griffiths method that is also very often used. 5 replicates per method, and all from the same sieved, mixed and frozen composite soil sample. And it was really frozen as like identical aliquots. So here, again, with LHPCR community fingerprinting method, you notice that at least for some of the methods, the replicates seem to cluster together and separate from other replicates. This was not really surprising. This was what we expected. Kits give you a little bit different result. This was more actually to test for DNA yield and for qPCR inhibition, because you want as much DNA as possible, because then it probably represents your community as well as possible, and you want it to be pretty clean, so that you don't have to dilute your sample horribly much to avoid PCR inhibition. Then that was the time when the MoBio kits recommended vortexing to break the cells for DNA extraction, and there weren't very clear rules like what kind of speed and time you should use if you wanted to use this more efficient beating lysis method for mechanical cell disruption. So then we tested different beating protocols. Again, we used the same kit, same soil, 5 grams, so pretty big, pretty representative soil samples. We used 2 machines, the Vortex recommended for this kit, and then we had this fast prep machine where you could also use 50 milliliter tubes. Tested 4 protocols. Just wanted to see, like, do we see any difference or not? Again, yield and integrity. You don't want your DNA to be broken into horribly small pieces, so that PCR would anyway work nicely. And you want nice yield. For the yield, vortexing gave the lowest yield, but also the most intact DNA. The other methods, in general, the higher the yield, the wider the band for DNA. so the more disintegrated DNA. So small differences in the bead beating speed can affect your DNA yield. This is for PCR inhibition. For the most efficient methods, when we did 3 times bead beating with a fast prep speed 4.0, if you put 1 microliter of template DNA in the PCR reaction, there was full PCR inhibition. If you used less template, or if you used the other methods, it amplified beautifully. And again, for community fingerprinting with LHPCR, you tend to see grouping based on the bead beating, so the cell disintegration efficiency. So what I'm saying is that if your bead beating machine breaks up and you plan to do rest of your samples with another machine, that's really not recommended. It can affect your results. So try to borrow somebody else's machine or get someone to repair it quickly. This slide I showed you already yesterday about the effect of template concentrations. So you want nice DNA yield to have a representative image of your community. But also what I would heavily recommend is that you use the same amount of DNA as template, or preferably also same amount of RNA as template, is if you do anything based on amplification, any PCR. I was talking about this yesterday, but we had for amplicon sequencing, for 16S-based analysis, we had 2 water samples. This was about 10 times as strong as the other one in terms of DNA yield as nanograms per microliter. These were actually supposed to be duplicate samples from an inoculant water in one experiment. And we were very surprised when we, in the first sequencing, saw very different microbial community compositions. They definitely were not good duplicates. So then we started looking at the template concentration and found out about this 10-fold difference. What we also saw is that when we diluted them more, so these are the different bacterial phyla, and these are the abundances of those bacteria from those phyla in the undiluted or non-diluted samples. And then when you put the same volume of DNA as template of 1 to 10 diluted sample or 1 to 100 diluted sample for the first water sample and then for the second. So what we found out that the template dilution really affects your recovered community. Some things that you see for both of these water samples is the decreasing relative abundance of actinobacteria in both of the samples, and actually really big differences upon diluting your sample. Same for Bacteroidetes, and for Proteobacteria you have an increase, a systematic increase for these 2 samples at least upon dilution. And for example here it's actually big, it goes from 38% to 66%. I would say that's a huge difference in the relative abundance of Proteobacteria. For metagenomics, of course you don't do 16S amplification, so should you not worry about template concentration at all? Well, at least according to one report from 2015. They demonstrate that DNA input level has a significant impact on community structure when you use assembly-based analysis. And this is caused by overrepresentation of low GC genomic regions following library amplification. So if you need to amplify your sample, you should also be aware that you normalize the input template amount. Another example for amplicon sequencing. This is from a phytoplankton community. My colleagues are trying to develop methods with which you can replace the microscopic evaluation or investigation of phytoplankton community composition and diversity with DNA-based methods. Anita, who made all these graphs, it's a recently submitted manuscript. She tested a mock community of known phytoplankton species, and they were always combined in the same proportions. So you had like really the same template for extraction from all of these replicates. These are the reference, what it should look like. The biomasses for the 6 phytoplankton species, the proportions were like this. The cell numbers were actually like this. This is the theoretical template ratio. This was done with, by the way, with 18S-based amplicon sequencing with phytoplankton suitable primers. And for pooled DNA it looked like this. But anyway, different kits. What do you actually recover? What kind of community? These are different DNA extraction kits and different RNA extraction kits. Huge, huge differences in the recovered community. The main and this is a mock community of pretty freshly cultivated cells. One of the main outcomes was that the RNA-based analysis actually gave a more realistic representation of the diversity than most of the DNA-based methods. But again, when choose your method and then stick to that. So the take-home message that I want and hope you will remember is that experimental sampling really makes or breaks your research. So try to ensure representativity and replication. And when you read, when I review articles or when I review, when I read reviewers' comments or reviewers' quotes, this lack of replication is really one of the biggest problems. So no fancy method can make up for lack of replication. And another take-home message, process all your samples in exactly the same way, because really surprising tiny things can affect your recovered communities. So those were my slides, and then do we have any comments, questions, discussion? We were actually already yesterday discussing a little bit, but it would be really interesting. Like now I have been talking about soil, mostly soil. Do you have any other cautionary examples or other things like from other systems? Actually heard about like human-based samples that for those dilution may actually help you to better recover the diversity. Would you like to actually share that with?
Okay, caught it. Yes, so I work with human microbial stuff, and our main source is fecal samples, where there's a lot of inhibitors, and in sequencing for also from biopsies, we've seen that inhibitors are a big problem when doing any type of PCR for sequencing or qPCR or anything. And they're often, after several rounds of purification with all possible methods, nothing but diluting the sample will be effective. But then that obviously compromises possibly your sample and the diverse set that you're going to get. But then again, we're not, in my work, interested in the very rare samples, but we want to see the whole community rather than just a few exceptions. So in that sense, it's different from the soil microbiota work. And also kind of to comment on the storage and the extraction, this is very similar to what we see. That then store everything in -80 or as cold as you can, and do everything exactly, and avoid doing things in batches that, you know.
Yeah.
Anybody else?
Any other? Now's the time to share your, like, because I've been so frustrated when seeing these results, and it's kind of nice to like share like all the time and money we have wasted. Like maybe it helps other to avoid the same mistakes. So if you have the same kind of advice, that would be great.
Okay, I have a question. If you're sequencing 16S, for example, you're interested in bacteria in soil, and you extract the total DNA. So that includes fungi, bugs, everything possible.
Yeah.
And like you said, that using the same amount of template for sequencing would be a good thing. But would it be good to, like if you're interested in bacteria, of course, the bacterial fraction might be different.
Yeah.
So what is your experience like? Do you quantify bacteria before you sequence and determine template for sequencing based on that, or do you just determine it by your total DNA concentration?
So if you are interested in also the quantities, and if you know that you have lots of other things than just microbes, like or microbial DNA, bacterial DNA in your example, then maybe ideally you would do qPCR with bacterial primers, and then based on that we your template. We have actually sometimes not for bacteria, but when I was doing TRFLP for sphingomoniads, I did that, and I got like better representation of sphingomoniads from different samples than when I just did amplicon sequencing, where they were like very little sphingomoniads in some samples and very much in some. So you could do that for plant microbiomes that we have been studying. We just use, if I normally use like from 1 to 5 nanograms of template for plant roots, we knew that most of it would be plant DNA, so then we just decided that okay, we use 10 nanograms, so tenfold the amount that we would normally, but there we didn't do 16S qPCR beforehand. So
can I comment something?
yeah, please.
So if you do 16S or any DNA, I think we don't measure the DNA. We do not try to have the same amount of DNA, since there is no method that you can say just by measuring the DNA will work in the same way in the PCR.
Yeah.
Since you don't know, like in skin samples, perhaps 99% of the DNA is human origin, and you don't know how it's going to work. So
Yeah.
the biological activity of the DNA, it's impossible to see. So we do it empirically. So we test with the first and the second PCR which of the samples will be the amount of that you can use.
Yeah, maybe it's easier because we often do like experimental settings, like experimental incubation, so we have the same soil type that we want to compare. So then it's probably much easier to to like it's more similar kind of samples.
With diverse samples and new samples, I think at least in our hands, it doesn't